Evening, guys. I hope so. You would have already seen the transcription and replication. Then we have talked about the genetic codons and mutations. And I'm very sorry I couldn't take this translation topic immediately after the codons due to some work. So today we are going to deal with the translation. So very, very important topic, translation. I am going to deal with it. So remember, the central dogma of your nucleus is going to function from a DNA. What it does is it tries to replicate another DNA, and from that what it does is it tries to make a RNA, or specifically mRNA, and this mRNA is going to produce protein. Okay, this mRNA is the one that helps in synthesis of protein. So there are different steps that are involved in this. That is what, from DNA to DNA you have your replication, from DNA to mRNA you add a transcription, then from mRNA to your protein, it is going to be a translation. It's going to be translation. This you need to remember. Okay, three things, guys. Remember, first you add a replication, then you moved on towards a transcription, then finally you add a translation. So these are the three important steps or three important process that occurs in the central dogma. And today we are going to describe about this particular one, that is going to be your translation. Shall we move into this? Do you have any doubts? No, right? Sure. Okay. So today we are going to discuss about the translation. So to begin with, I hope so you all remember this one particular topic which we have already discussed, that is going to be your charging of your amino acid. I think how many of you saw the previous video that is on codon, I don't know. But in this codon video, I have discussed about this one particular topic that is going to be your this one particular topic that is going to be your charging of your amino acid. So what happens is, basically at the end of your transcription, you're going to have mRNA. This mRNA is going to be coded with a certain sequence. Okay, that is required for the synthesis of new protein or new amino acid or new enzymes or so. All the building blocks of our body is going to be from this process. Okay, from your gene, from your DNA, RNA is formed, mRNA, and this mRNA consists of codons that is going to be helpful in the formation of proteins and enzymes. So how is this codons in your mRNA is going to be helpful in producing a protein? So that is where comes, apart from this mRNA or messenger RNA, you have two more RNA, that is going to be your transfer RNA and your ribosomal RNA. This transfer RNA, I think I have discussed very detailed in the previous session. So what happens basically is, you are going to have this tRNA, and this tRNA is going to have an anticodon on an amino acid acceptor arm. So that side it's going to be amino acid acceptor arm, and down it is going to be anticodon arm. What happens is, there are 20 different okay, this is the normal structure. First I'll tell like this. This is your first normal structure of your tRNA. Now this tRNA is going to be carrying certain amino acids in order to code them and in order to produce that protein. So what happens basically is, first you're going to have a tRNA separately. This tRNA is going to be loaded with one amino acid. This is the first step, charging of your amino acid, charging of your tRNA with amino acid. That is going to be done by the enzyme called as amino acyl tRNA synthetase. What happens in the amino acid tRNA synthetase enzyme? Listen carefully. So you have a tRNA, guys. In this tRNA, there is another molecule coming and sitting at the amino acid acceptor end. So this can be methionine, this can be arginine, this can be any phenylalanine, tryptophan, all the essential amino acid. Why we call this as essential amino acid, guys? Because this amino acid has to be intaked from your dietary in order to produce this protein, So that is what exactly happening. So you eat amino acid, this amino acid has to be converted into a protein. But how does this protein conversion occurs? That is going to be occurring by this translation method. So you have a tRNA, guys. This tRNA is going to be loaded with all the essential amino acid. So what are the number of essential amino acid you have? 20 essential amino acid. That is the reason there are 20 different isoenzyme of this amino acyl tRNA synthetase. So there are 20 different amino acid tRNA synthetase. Depending upon what amino acid you wanted to load over your tRNA, the enzyme is going to differ. Clear, right? Till this point, do you have any doubt? You can put in the chat box. Let me move towards the next important step. So now you are going to have a tRNA loaded tRNA loaded with the amino acid. This is going to be the first step. But what happens is there is actually a utilization of ATP in this step. So ATP is converted into AMP. Whenever ATP is converted into AMP, there is going to be 2-phosphate is going to be released. If you are going to consider 2-phosphate release, that means that here you require 2 ATP. So it is equal to spending 2 ATP. Therefore, what you need to understand is here you are going to spend 2 ATP. Here you are going to spend 2 ATP. So at the end of the day, at the end of the discussion, I'll be asking you to calculate the total amount of energy spent by this translation process. At that time, I need answer from you all. Is it clear, guys? Is this clear? Shall we move towards the next one? So remember, once this step is over, okay, this step is over, this step is the place where there is a point of proofreading. So only at this particular step you can proofread. Once your amino acid RNA is synthesized, once it is going to go and sit over your mRNA, once it is going to transfer, once it is going to produce a protein, you cannot change it. So the steps which are going to come forward, they are going to be irreversible. That is what I want you all to understand. Once after you guys understand this, next we are going to move towards initiation. So basically what happens is your amino acid, okay, your translation step is going to consist of 4 important steps. What are the 4 important steps? Along with me tell. Number 1, activation of amino acid or charging of amino acid by tRNA. The second step is going to be, as usual, like you saw in your transcription, you're going to have initiation. So now like that, you're going to have initiation, then you're going to have elongation, then you're going to have elongation, then finally the 4th one, that is going to be your elongation, and then finally you're going to have termination. So these 3 are similar to that of your previous step, that is going to be your transcription. Same way, in your transcription, I hope you all remember that even in your transcription, for all these steps to occur, that is going to be your initiation, elongation, termination, we need certain enzymes. You saw that. So we saw different types of enzymes. If you remember, I don't know, I'm not sure. If you remember, just recall and tell me that you need different enzymes. What are the different enzymes? You need RNA polymerase, you need a Grey factor, you need a termination row dependent, row independent. Same way we have seen in your transcription, isn't it? Same way here also, for each and every step, you will be requiring different enzymes. Different enzymes or different factors are necessary. So what are those factors? For initiation, we are going to have initiation factor. For elongation, we are going to have elongation factor. Similarly, now itself I tell you, same way, prokaryotes, it is going to be different, and in eukaryotes, it's going to be different. In prokaryotes, the initiation factor is just named as IF. Elongation factor is termed as EF. Whereas in your eukaryotes, you're going to adjust small e to it. So initiation factor, you call it as EIF. Elongation factor, you call it as EEF. So E small e refers to that of the eukaryotes. So these are the 2 things which I want you all to remember. Once you remember those things, next what you're going to remember is, I told you that there are certain sequences. So your DNA lende உங்களுக்கு RNA produce ஆகும்போது உங்களுக்கு எந்த sequence produce ஆகும்? Do you remember? Tata box, cart box. Similarly, the mRNA, where your codons are going to be there, where you're going to start your synthesis of your proteins, that sequence, you call it as a Shine-Dalgora sequence. There are 2 important roles in your Shine-Dalgora sequence. Remember, guys, always in any human activities, the first amino acid produced is going to be your methionine. So any sequence, it starts with AUG. Similarly, the termination occurs with the nonsense codons. What are the nonsense codons? UA, UGA, UAG. That is going to be UAG, UA, UAA, and then UGA, UGA. So these 3 are the stop codons. These 2 are clear, right? Yes. So now what happens, basically, I'll show you the slide also. First, listen carefully to this diagrams, then we will see the slide, okay? Then I will show you an animation. So three structured we are going to learn today. So you have this amino acid accepted tRNA. We call it as amino acyl tRNA. Now what happens is, once you have amino acyl tRNA, remember in eukaryotes it is going to be methionine loaded tRNA. That is the first formed amino acid. Okay, first formed amino acid by your translation. But unfortunately in the prokaryotes we call it as formyl methionine loaded. Therefore the first amino acid which is going to be seen in the proteins in your eukaryote, in your prokaryotes, it's going to be formyl methionine. Two important things. Initiation phase is going to be always methionine or formyl methionine. Termination is always UA, UGA, UA. Okay, these two things you need to understand. Once you understand this, next important thing what I want you all to know is, let it be methionine or formyl methionine. What is the codon? What is the codon? Codon is going to be AUG. If the codon is AUG, what will be the anticodon end of it? Anticodon is going to be UAC. Therefore, the anticodon which is going to be present, the codes which are present in the anticodon end of your tRNA is going to be UAC. Now, what happens is you have a very big sequence. Okay, you have a very big sequence that we called it as a Shine-Dalguara sequence. Now, what happens? First, your mRNA comes and sits on your your tRNA, that is amino acyl loaded tRNA, methionine loaded tRNA, or formyl methionine loaded tRNA, depending upon prokaryotes or eukaryotes. It is going to come and sit on your mRNA. So they are going to come and sit on the mRNA. They are going to search this one particular methionine AUG. Once it is going to got that particular code, it is going to come and attach at that point. But before that, what you need to understand is, these two are going to be separate molecules. There has to be someone who is going to bind both your mRNA and both your tRNA and start the translation. That's where your ribosomal mRNA, ribosomal RNA comes into action, or RNA. Do you know that this protein formation or protein synthesis occurs in the ribosome? We usually read. Why do we read like that? Because ribosomal RNA present in the ribosome, that is going to start the initiator. That is going to initiate your translation. How do they initiate? That is the next step we are going to see. That is the initiation phase. In initiation phase, what you need to do is, you need to make sure that your tRNA, okay, your mRNA and your ribosomal RNA, all three are grouped together to form a complex. How do they get grouped together to form a complex? So remember guys, in your, you have your so-called your big one, that is going to be your larger unit and a smaller unit. In a prokaryotes, it's going to be 50S, 30S. In eukaryotes, it's going to be 60S, 40S, isn't it? So that's all we learned in the last class. Remember, what you have is, you have three important sites. You have three important sites at your ribosomal RNA smaller unit. So you have larger unit, smaller unit. First, what happens? The smaller unit and larger unit are going to be separated. So first, what happens is, your larger unit and smaller unit, they are going to be separated. Larger unit and smaller unit, they are going to be separated. So like this, they will be there. Okay, first they are going to be separated like this. They are different, separate unit. Okay, remember, they are going to be separate unit like this. Okay, they are going to be separate unit like this. That's what I want you all to remember. What happens is, first there is going to be a tRNA. So first what you have is, you have this 30S smaller subunit. Then this 30S smaller subunit, you don't have tRNA also. First just erase the tRNA also in your mind. Now you have a separate ribosomal smaller subunit alone. This smaller subunit consists of three important structure, three important finger-like projection, or three important sites. What are those sites? A site, P site, E site. I want you to remember this order. Okay, ape. Remember ape in the reverse direction. APE, ape. So you have a ape. Okay, so A stands for acceptor site, P stands for peptidyl site, and E stands for exit site. Okay, so you have three sites. Now, what happens is, first you are going to have a important protein or important factor. You have three factors like initiation factor, 1, 2, 3. All this I'm going to talk about prokaryotes first. So the first one you wanted to know is, you have a initiation factor. What is that initiation factor? You have an initiation factor. This is your initiation factor. This initiation factor is nothing but they are going to be initiation factor 1. you are going to have an initiation factor 1. Do you remember now? Is it clear? First, you have an initiation factor 1. This initiation factor 1 binds with the A side. So such that the incoming, now just remember, I told you, don't, this diagram, the tRNA நடுவுல இருக்குல அதை மட்டும் மறந்துருங்க. The tRNA இல்லன்னு நினைச்சுக்கோங்க. Okay, now you have a A side which is going to have initiation factor 1. Next, this is going to prevent the incoming tRNA to go to the acceptor side. அப்ப ரிமைனிங் இருக்கிறது உங்க பெப்டிடைல் சைட் டு எக்ஸிட் சைட்டும். கரெக்டா? எக்ஸிட் சைட்டுக்கு யூஸ்வலா எதுவுமே போகாது. காம்படிஷன் இஸ் பிட்வீன் வெதர் இட் ஹாஸ் டு கோ டு அக்செப்டர் சைட் ஆர் இட் ஹாஸ் டு கோ டு த பெப்டிடைல் சைட். இப்போ அக்செப்டர் சைட்ல இனிஷியேஷன் ஃபேக்டர் 1 இருக்கிறதுனால இட் வில் பி கோயிங் ஃபார் த பெப்டிடைல் சைட். அப்ப வர்ற அந்த டிஆர்என்ஏ ஒருத்தர் கைட் பண்ணும். Okay, which one has to go to your peptidyl side? Someone has to guide it. So who is going to guide that? The guidance is going to be from your initiation factor 2. So you have another factor that is called as initiation factor 2. Both of them, what they will do is they will make sure that the incoming tRNA comes and attaches with your P side. Okay, so now your incoming factor is going to come and attach to the P side. So now you are happy. But in between this process, you have to make sure that the larger subunit does not come and attach with the smaller subunit. So that one has to be prevented by someone. E is nothing but your initiation factor 3. Is it clear? Now, all these processes occurred. Your tRNA came and sat on your amino acid loaded tRNA. This is amino acid loaded tRNA. This came and sat on your peptide side. Okay, very clear. That's all. So now what happens is slowly the first your initiation factor 3 is going to be removed. So like this, first what happens, your initiation factor 3 gets removed. Once initiation factor 3 gets removed, your initiation factor 2 and initiation factor 1 also should get removed. For this removal of initiation factor 1 and initiation factor 2, what do you need? You need a GTP. So here what happens is 1 GTP gets converted into a GDP. 1 GTP gets converted into a GDP. So see here how it does. So 1 GTP gets converted into GDP, and once your GTP gets converted into GDP, what happens? Your initiation factor, all 3 initiation factor will be leaving the place. Once all 3 initiation factor leads, it's going to be moving out of the place. What happens? Your 50S ribosomal RNA, see here, your 50S ribosomal part of your RNA comes and attaches again in the your subunit, subunit of 30S. Therefore, it is going to form a initiation complex. This is 50S, 30S. Together, we call it as 70S initiation complex. Remember? That's all. Do you have any doubt? Same way, same step is going to occur in your eukaryotes also. But the difference between your prokaryotes and eukaryotes is, in prokaryotes, you have initiation factor 1. Here, you are going to have eukaryotic initiation factor 1A that does that work. And you have initiation factor 2. Here you have eukaryotic initiation factor 2 and eukaryotic initiation factor 5B. Same way there you have initiation factor 3. Here also you have eukaryotic initiation factor 3. So do you remember? Now you can understand the 3 ones. Okay, now you understood all the 3. Okay, now in addition to it, what is the difference between your prokaryotes and eukaryotes is, here you are going to have a initiation factor 4. What is the role of initiation factor 4? This initiation factor 4, which is going to also consist of, okay, it is going to consist of 3 subunits. What are the 3 subunits? A, G, E. Reverse it. Okay, A reverse panno, inge age reverse pandrenga. So you have AGE. These 3 are the subunits of 4F. Ipa latest edition of Harper they had one more subunit. In the latest edition of Harper, they had one more subunit, that is B. So what are, now tell me, what are the 4 subunits of your initiation factor 4? There was one question in your AIMS exam once. They didn't give anything else. So they're simple. The question was, which of the following are not part of IF4F? IF4F. This is what they gave. Many people got confused. What is this IF4F? First, you have to know what is IF4F. Then you have to know what are the components of IF4F in order to eliminate, rule out the one which is, which are the following is not. You have to know which are the following is not. They gave option like A subunit, G subunit, E subunit, F subunit. And second time, the same question came in the NEET exam. That time how they asked is, the same question, the options were A, G, B, and M. So that time what happened is B was not, many people didn't notice B was added in the R pair, and what they did is they didn't select they selected option B. Okay, so they got confused between G and B, M and B. So many people had a 50% split answers. So but B is also now included as a part of IF4F. Okay, so the same process occurred and you got a 80S initiation complex. You got a 80S initiation complex. So now tell me, what are the stages of translation? So first you have your initiation factor 1, 2, 3. They form 30S initiation complex. Now what happens? There's your GTP comes and cleaved into GDP, and all your factors are moved away to form this very important step that is going to be your, what is that? That is going to be your 80S initiation complex or 40S initiation complex. Now you got it. Now what happens? First, what you have to do is you need to bind your tRNA. Now I told you how your tRNA is coded. So tRNA is going to read your mRNA framework, and once it reads your mRNA framework, it is going to find at what place it has to go and sit. Before that, you have to remember that once you are the one which has come and sat is not going to do any work. Okay, the one which has came and sat on your methionine AUG initiation factor, it is not going to do anyway. Now the second work, what happens is, see here, now you have this framework, isn't it? Now what happens is there is going to be another tRNA loaded amino acid. There are now you have 20 different tRNA loaded amino acid. All of them are going to come and check whether I am suitable for this. This is like a lock and key model. So everyone are going to see whether they are the key for the lock which is present as a codon. Okay, out of the 20, one of them is going to, out of the 20, one of them is going to code for the amino acid that is required in your protein. So in a protein, the first one is methionine. After that, you can have arginine, or you can have valine, or you can have tryptophan, you can have phenylalanine, whatever it is. So you have such a long thing that you need to understand that someone is going to come and sit at that place. Who is going to come is going to be depending upon. So who is going to come is going to be depend upon. Now what you need to understand is, once you understand all these things, once you understood all these things, next what you are going to do is, what you understand from this. So there are going to be 20, it's going to be a many amino acid loaded tRNA is going to be coming and going like a lock and key. At last, what happens? One amino acid, probably they will be like having the same and codon. Okay, same anticodon similar to that codon present in the mRNA. So your tRNA would anticodon, mRNA like codon match agudu. Once they get matched, what happens is it is going to come and sit on this. So in the madri occur. So now see here you have APE. So once the you have formyl methionine or methionine which is now present in your P site. Okay, A site new one. Let me like give an example, arginine. So arginine oda idu vandu AGG. So adukku similar UCC irukku ungaloda anticodon. So they have come and attached at this particular site. After coming and attaching, so in your after coming and attaching, there are some factors which are involved similar to that of initiation phase, but I will discuss it at the end because it will be very confusing. Now what you need to remember is, you have a site like this, APE. At this APE site, so what happens is your first in the peptide, the ribosome is going to move along the direction. So first ungaloda methionine site la P வந்து attach ஆயிடுது, அது ஓகே. இப்போ அடுத்த frame shift க்கு போகுது. So அடுத்த shift of codons க்கு போகுது. So for example, first you add this AUG இல்ல, now there is going to be having AGG. இந்த AGG ல வந்து உங்களோட arginine loaded tRNA is going to come and sit. வந்து உட்கார்ந்தோனே next step என்னன்னா, bind ஆயிடுச்சு tRNA. It has read the mRNA framework. After reading mRNA framework, the third important step is peptidyl transferase activity. So you have a enzyme called as peptidyl transferase. Okay, you have a enzyme called as peptidyl transferase. Okay, this peptidyl transferase is present in the RNA. It is going to present in the ribosome. So 23S ribosome, rRNA ribozyme. So it's a ribosome produced enzyme. That's why we call it as ribozyme. So it is called as ribozyme. So it's a 235 rRNA ribozyme. Sorry, 23S rRNA ribozyme. So this 23S rRNA ribozyme is going to cut the amino acid in the tRNA. So in the amino acid la irukkudiya the P site la irukilla amino acid. And the P site la irukkudiya amino acid la irukra and the amino acid P site la irukra tRNA la irukra amino acid. Again, I'll repeat in English. So you have a tRNA in your P site. That P site there is going to be a tRNA, right? Along that you are going to have amino acid loaded. Now what you're going to do is you're going to cut this amino acid from your tRNA present in the P site, and you're going to transfer it to your A site. So this transfer requires a GTP again. So what is this process called as? The transfer of amino acid from your P site to your amino acid tRNA at your A site because of the action of peptidyl transferase is called as translocation. This translocation is going to use one more GTP. So at the beginning, we used one ATP. Then in the first step, we used one GTP. Now in the second step, you're going to use another GTP. Okay. So this are 2 GTP and 1 ATP you used till now. Okay. Now what happens? This is going to go away. Okay, this is going to go away. Now what you need to do is you have to shift the tRNA. You have to shift the tRNA. Okay, you're going to shift the tRNA from A site to P site. So in the translocation, எப்படி நடக்குதுன்னு தெரியுமா? Translocation not exactly just amino acid transfer, but there is one more thing. What is that? The one more thing is going to be the movement. Along with this, what happens? There is going to be movement of ribosomal RNA on mRNA. For this process to occur, what you need is you need a elongation factor G. You need elongation factor G. This is going to be in relation to your prokaryote. So what you need? You need a elongation factor G. Up to this, is it clear? Okay, so what is translocation? I told you, once there is a movement transfer of amino acid from P site to A site, how it does is your ribosome is going to move. So your ribosome in the direction la move agudana, what happens? This is going to go to exit site. This is going to come to P site. Okay, this is going to go to exit site and this is going to go to P site. So in the my movement agumbodhu, what happens? Automatically when your A site la irundha amino acid, what happens? Now it is going to amino acid loaded tRNA. It is going to go to P site. Now you have methionine. Now you have arginine. In a prokaryotes, formyl methionine and arginine. This is going to be same in the both prokaryotes and eukaryotes. For this to occur, in your eukaryote, you have 4 different factors. What are those factors? Very similar. So you are going to have a EF 1 alpha, 1 beta, 1 gamma. You have EF 1 alpha, 1 beta, 1 gamma, then F2, EF2. 1 alpha, 1 beta, 1 gamma, and EF2. This is elongation factor. Similarly, prokaryotes are going to have 3 things. Number 1, elongation factor TS, elongation factor TU, then you're going to have your G. G is the one that is required for shifting of the peptidyl transferase amino acid from your P site to A site. Till this, is it clear? So till this, just understand how many ATPs and GTPs are utilized. Till now, we have utilized 2 ATPs for activation of your amino acid and 2 GTP, 1 GTP used at the entry site and 1 GTP used in the translocation. So you need 4 high energy phosphate for addition of 1 amino acid in the growing polypeptide chain. Similarly, for each amino acid add-ons, you will be utilizing 4 ATPs. This is what I want you all to remember at the end of this discussion. Once you understand this, what happens? So now what happens? You're going to have like this. Okay, so you're going to have a shift like this. Just understand this alone. Okay, I'm just copying the same thing. You can also just redraw this again. This is very important. So now you understood that you add this one particular thing. Okay, now you add this one particular thing. This is going to slowly move and it is going to go to the termination site. Similarly, you're going to have 3 important things. Okay, these 3 things is going to be, again, it's going to be placed at the same right. So then what happens? This you're going to have, this is going to go and move like this. Okay, then you're going to move like this, and you also going to have big, this is going to be your bigger, small upper unit. So what happens is now you have moved on your code towards the end, that is going to be called as termination site. Now, can you tell me what is the codon for your termination site? The codon for your termination site is going to be your nonsense codon. What is the nonsense codon? Either it is going to be UA, UGA, it's going to be UA, it is going to be UGA, it is going to be UAG, it is going to be UAG. You have three termination codons. So what are the three things? UA, UGA, UAG. So you have three important things. Okay. Out of these three important things, what you have to understand is, see, this is going to be your mRNA. So you're going to have like this, you're going to have very big, now you have a very big amino acid loaded. So like this, you have different amino acid. And what is the first one? Methionine is the first one. So like this, you have amino acid. Now you need to cut that amino acid. You need to cut it away. So that is where you're going to have the releasing factor. So that's where you're going to have releasing factor. Now you're going to come to the end of it, releasing factor. So you have different releasing factor actually. So what you need to remember is you have different releasing factors. So though the name tells releasing factor, the name indicates that it helps in release. However, it does not help in release, but it recognizes a stop codon. That's all. Once you recognize a stop codon, it is going to be releasing itself. So அதுவே வந்து வெளில போய்டும். ஓகேவா? அந்த releasing factors என்னன்னா RF1, RF2, you're going to have RF3. இந்த மாதிரி prokaryotes இருக்கு. Okay, whereas in eukaryotes, you just have eukaryotic releasing factor. This is going to be in the eukaryotes. This is going to be in the eukaryotes. Is it clear, guys? That's all. This is what you need to understand. That's all. Okay. Hope you all understood. Okay. So in translocation in prokaryotes, I told you it is going to be done by EFG. Who is going to do in eukaryotes? In eukaryotes, it is going to be done by eukaryotic elongation factor 2. Eukaryotic elongation factor 2. Okay, that's all. That's what you need to understand. So once it is going to go to the terminal side, what happens? It's automatically going to cleave it away and it is going to go away, and you have a very beautiful enzyme or protein structure which is going to come out of it. So this is what you all have to know. Okay, this is what you all have to know. So once you understand that, okay, once the releasing factor is going to identify, once your releasing factor is going to identify it, then what happens? You are going to be producing a protein. So now at the end of the day, what you need to understand is how many ATPs are produced, how many high energy phosphates are used. 4 high energy phosphates are used. Is it clear? Now what we will do is we will just see a video. And I'll explain you carefully same thing. After actually understanding that video, what we will do is we will again come back. We will again discuss next important topic, that is going to be the post-translational modification. Can you see the video? Hope the video is visible. Just I'll give you the commentary. Just listen carefully. What happens is, in a translation, I'm just starting from here. You're going to understand the structure, function of the ribosome, understand the function and charge tRNA, then understand the translation, initiation, elongation, and termination, what happens in the ribosome. You all understand that, you all know that in your ribosome, you're going to have ribosomal RNA. It is going to consist of 60S and 40S subunit in your eukaryote, 30S and 50S subunit in your prokaryotes. You know that from the DNA, you did a transcription, you have got a mRNA. After you obtaining a mRNA, what you do is, you're going to have a ribosome. Now what happens? You have a mRNA, you're going to have a ribosome, you're going to have a charged tRNA and uncharged tRNA. So you're going to have utilize different factors in a prokaryotes. Initiation factor 1, 2, 3. Along with it, you have G factor, TU factor, and EFTS factor. Then you have releasing factor 1 and releasing factor 3. Okay, then what happens is, first your ribosome reads your messenger RNA. It is going to directly synthesize the encoded protein. So it is going to start at the AUG and it is going to end at the UAA, UA, UGA, and UAC. Now you have to understand that there's a 2 larger subunit and smaller subunit. In prokaryotes, you have this 30S and 50S. Old ribosome is 70S, and in eukaryotes, you're going to have 40S, 60S. Old is going to be called as 80S. You have the Svedberg. What is the Svedberg? It's a unit of sedimentation velocity. Sedimentation is ultra centrifuge, depends on both the mass and and shape of a molecule. It's not simply a measure of molecular mass alone, but it takes the molecular mass as well as the shape into consideration. Next, what happens is, what you need to understand is, ribosomal subunit is going to assemble and disassemble again and again. And this is called as a ribosome cycle. You know that 4 phosphate is required, 4 high energy phosphate is required for this ribosomal cycle. So this is your start. So start, you're going to have AUG, then you have GCC and etc. Now what you're going to understand is, so always triplet codon, you call it as triplet codon, always immediately adjacent. So AUG and GCC. Now what you need to understand is, you have 3 important structure, A, P and E. First, what happens is, this amino acid loaded with a formyl methionine in your prokaryotes, it is going to come and sit on the P side. Who is going to prevent it from going to, let's say, A side? Your elongation factor, initiation factor 1. So you have this amino acid tRNA acceptor side, you're going to have peptidyl P side, and E is called as your exit side. So there is a tunnel that is going to allow the growing polypeptide to exit ribosome. Then you have this translation initiation. This is going to require your ribosome bond to the mRNA. Then ribosome is properly aligned over the start codon. Then the P side of your ribosome containing charged tRNA. So these are the 3 things are the component of your initiation phase. So you have 3 factors: initiation factor 1, initiation factor 2, initiation factor 3, and you know all the functions of your all the initiation factor 1, 2 and 3. Can you see here? Initiation factor 3 sits and it prevents the binding of both the 50S and 30S, and you're going to have initiation factor 1, initiation factor 2 along with your GTP. Now what happens? Your initiation, your tRNA loaded with a formyl methionine in a prokaryotes, methionine in eukaryotes is going to come and sit on the P side. So it prevented from entering into A side. Now what happens is, you have the Shine-Dalgarno sequence, base pair with the 16S rRNA on the ribosome. I told you already this one. So this is called as 30S initiation complex. Once this is formed, what happens is, you're going to remove all the initiation factors 3, 2, 1, 1. So while removing this all initiation factors, what happens? Your ribosome comes and sit on it, and it's going to form the 70S ribosomal 70S initiation complex. So you have this GTP. What happens? The GTP is going to be removed to give out GTP, and now it is going to be starting to move. So now initiation process is over. Now it is ready to produce synthesizer, and it is ready to accept one more tRNA loaded with amino acid in your acceptor side and start the translation. So now what happens? You need to understand there is 2 things that are going to be different from your prokaryotes and eukaryotes. So that I already told you everything. So in a eukaryotes, what happens? Same way, initiation factor 1, initiation factor 2, initiation factor 3. In addition to it, so you're going to have initiation factor 2 and 5B together. So it is going to be having your GTP. So now you're going to have GTP loaded on it. So what happens? It is going to be sitting. Now you have 3 important structure. I told you, right? Age. So that is your 4F complex. This 4F complex is going to be preventing the initiation. Once this 4F complex is going to be removed from your 5' cap. So what happens is, it's going to be producing. It is going to be, you're going to have B that is also going to come and attach. So once B comes and attach, it is going to be moving that. So it is going to be moving that. Once that part is going to be removed, once EGAF part is EGAB part is going to be removed, what happens is, you are going to have now can you see? Now what happens is, so EGAB is going to be slowly moving, and now slowly what happens? There is going to be a beginning of your translation. So now your ribosome is ready to begin the protein synthesis. Once your ribosome is ready with protein synthesis, you're going to have elongation. So for elongation, what do you need? What do you need? You need a tRNA, another amino acid loaded tRNA. This amino acid tRNA is going to be coming and sitting on the acceptor side. So once it sits on the acceptor side, what happens? You're going to have transfer of your amino acid. Okay, there's going to be transfer of amino acid. Who is going to do? Peptidyl transferase is going to do. Peptide transferase, once it does, your ribosome moves, and the tRNA which is present on the A side is going to coming and sitting on the P side. For this, who is going to reclaim? You're going to have some elongation factors. What are the elongation factor in a prokaryote? It is going to be TS, G and TU. In eukaryotes, you're going to have alpha, beta, gamma and 2. So what is tRNA accommodation? It is a process that ensures correct pairing between the tRNA and your mRNA. If there is an incorrect pairing like this, so what happens? If there is an incorrect pairing, so it is going to be removed. So only the correct pairing can be sitting on that place. This prevents the mutation. So that's why I told you that the first factor, first one has to be correct. So once if that is going to be correct, every process that is going to be occurring adjacent to it after that, it's going to be correct. So this is what I told you. Next, what happens? The amino acid tRNA is escorted. Who is going to bring that amino acid tRNA to your mRNA? That is going to be the role of your elongation factor TU. What your elongation factor TU does? It does a escorting work. So once that is done, so there is going to be another GTP which is going to be used at this particular site. There is another GTP that is going to be used particular site. So your ribosome moves one codon down mRNA. So in the movement GTP தேவைப்படுது. So it's looking like நீங்க அந்த கவுண்டர்ல வந்து when you are going to buy something in the supermarket, so in a very big supermarket, what happens, there is going to be movement of this. So அடுத்த பொருள் வந்துட்டே இருக்கும் அப்படியே பில் போடுறதுக்கு. அந்த மாதிரி அந்த மூவ்மெண்ட் பெல்ட் இருக்குல்ல, அந்த பெல்ட்டோட வேலையை தான் உங்க mRNA ல ரைபோசோம் ஆக்ஷன் ஆகுது. So you're going to have இந்த TUG எல்லாம் என்ன பண்ணா ரீசைக்கிள் ஆயிட்டே இருக்கும். GUG எல்லாம் வந்து it is going to continuously get recycled. It's going to continuously getting recycled. This is what you all should remember. Once that is going to get recycled, what you have to understand is you're going to have the stop codon signal. So stop codon signal you're going to have UA, UGA and UA, UGA. So they are going to be termination. This is going to be same in case of prokaryotes, it's going to be eukaryotes. But what you need to understand is you have this different factors, releasing factors, you have different releasing factors. RF1 recognizes all the stop codons in eukaryotes. In prokaryotes have different RF codons. So the class 1 release factors bind to the A site and facilitate the release of your protein. So one completed protein release once there is going to be a release of the completed protein. So what happens? Class 2 factor release the factors and recycle the class 1 release factors. So they're going to release the class 1 recycle factor, and like this is going to remove the removed polypeptide chain. It is going to go and it is going to be synthesizing new protein. Till now, is this clear, guys? Do you have any doubt till now? If you don't have any doubt till now, what we will do is I'll just move and I'll just discuss shortly about how to write in your exam. I'll tell you how to write the same thing, whatever we discussed, how to write in your exams you're going to see. Okay, you have, so how to write in your exams, let's see. So first you're going to discuss about translation. So you're going to write the definition of translation. This PDF I'll just send you. Translation is a process by which the genetic information present in the form of sequence of bases in your mRNA is expressed to produce a sequence of amino acid in your protein. And this decoding of your mRNA into a protein is called as translation. So there is going to be a different phases, as I told you. Now you have activation of amino acid. So once there's activation of amino acid, there's an initiation of protein synthesis. So you're going to have formation of the protein synthesis complex. Same video, whatever recorded in that video, whatever told in that video, I've given the pictures as a PDF. I'll just send you, you can just take it. So then you're going to have elongation. Once you know this elongation, once you understand this elongation, then what happens? There's a termination. After termination, what you need to have is you need to know the stages of translation, everything given in a one slide. After that, you're going to have a polyribosome. What is this? Many ribosome works on the same RNA molecule simultaneously, and it is going to aggregate to form a polyribosomes. So you have this polycistronic mRNA. More than one polypeptide synthesis from one single mRNA can occur. That is called as polycistronic mRNA. So what are the energy requirement? Remember, 2 initial activation of amino acid, 2 in the elongation peptide formation, 1 ATP in initiation complex, 1 GTP in ATS ribosome formation, 1 for termination. So what happens is just forget all these things. Okay, I'll just give you now currently what your energy expenditure, what is going to be the energy expenditure that is going to be given in your ARPR is, okay, you need only 4 high energy peptide bond, 4 high energy peptide bond. Peptide, okay, for 4 high energy phosphate, 4 high energy phosphate, it is going to be obtained from 2 from your ATP, 1 from your GTP, 1 from your 1 from your GTP, okay, at the initiation phase, and 1 GTP at the elongation phase. So முன்னாடி கான்செப்ட் வந்து டெர்மினேஷனுக்கு ஒரு GTP யூஸ் பண்ணனும்ட்டு இப்ப அந்த டெர்மினேஷன் GTP எல்லாம் யூஸ் ஆகிறது இல்ல. So that's what I want you all to remember. Next, you are going to move towards a post translational modification. What is the post translational modification? There are some reversible and some irreversible. Just write like this. Okay, just write like this, whatever I am writing. Post translational modification. Just write the heading as post translational modification. Okay, just put the heading like this. So what happens is you have 20 amino acid, 20 tRNAs. So pre-translational modification, it is going to form 20 amino acyl tRNAs. So then it is going to form nasal peptides, not nasal polypeptides. You know, right? This nasal polypeptides undergo certain post-translational modification. So it is going to undergo certain post-translational modification. I'll tell you. So எல்லாருமே post-translational modification சொல்லுவாங்க. But I am going to tell something different that is called as co-translational modification. So put the heading as translational modification. Just write this 20 amino acid, you're going to have and 20 tRNA you're going to have at the beginning of this. So what you're going to have is, at this particular phase, there can be pre-translational modification. Will you accept it? It is called as pre-translational modification. Translational modification. Afterwards, what happens? Modification. After that, what happens? You're going to have 20 amino acyl, 20 amino acyl tRNAs. You're going to have 20 amino acyl tRNAs. This 20 amino acyl tRNA, it is going to form nascent polypeptide. At the end of your translation, you're going to get one, right? That is called as nascent polypeptide. Nascent polypeptide. So that is called as a co-translational modification. So in the process நடக்கிற modification இருக்கு, அதுக்கு பேர் co-translational modification. Okay, then you are going to have a nascent polypeptide moving into complete, or you can call it as a mature polypeptide. Mature polypeptide. Okay, so that is going to be called as post-translational modification. Post-translational modification. Okay, so once you got this post-translational modification, so now I'm going to talk about this so-called co-translational modification. What is this co-translational modification, guys? Remember, this occurs in case of selenocysteine and pyrrolysine. So you're going to have two amino acids. Number one, selenocysteine, which is present 21st amino acid, and you're going to have this pyrrolysine. Pyrrolysine is going to be 22nd amino acid. Okay, 21st and 22nd amino acid. So these are going to be the co-translational modification. Then what is your post-translational modification? Just write down post-translational modification. You are going to divide into post-translational modification. You are going to divide into two things. Okay, how we are going to divide? We are going to divide into reversible process, and you're going to divide into irreversible process. Irreversible process. So you're going to have two things, reversible, irreversible. Just remember, okay, what are the reversible factors? Okay, I usually remember like this: DGP and lump. DGP and lump. Okay, so D stands for disulfide bridge. Disulfide bridge. Okay, and G stands for glycosylation, and P stands for phosphorylation, and A stands for acylation, N stands for N-acetylation, and D stands for, remember, how do I tell this? D is nothing but ADP, ribosylation. So this is going to be your DGP, and L stands for your lysine hydroxylation. I'll talk about all these things. Lysine hydroxylation. Then you're going to have U. What does U stands for? What is ubiquitination? Ubiquitination, and M stands for methylation. And your P stands for proline hydroxylation. Then another P stands for proteolysis. Why this is important? Have you ever thought why all these things are important? See, you all read that there are 20 essential amino acids. Okay, from this 20 essential amino acid, all the remaining amino acids are synthesized. Now do you understand? So you have certain amino acid called as essential, you have certain amino acid called as non-essential amino acid. Essential amino acid has to be supplied from your dietary. From the amino acid that is supplied from your dietary, you're going to produce the remaining amino acid by following steps, either adding a disulfide bridge or glycosylation, phosphorylation, methylation, ubiquitination, lysine hydroxylation, proline hydroxylation. From all these processes, you are going to produce another set of amino protein. So that is exactly what you have to understand. So once protein is formed by a post-translational modification, your protein is going to undergo folding. So what happens is, once your protein is formed, protein is formed, what happens is, it is going to undergo post-translational modification, post-translational modification. And what happens is your proteins are going to undergo, it's going to undergo folding. Once your proteins undergo folding, this process is called as, it is going to be done by chaperones. So who is going to do that? It is going to be done by chaperones. Remember, who is going to do that? It is going to be done by chaperone. I want you all to understand this one particular point. It is going to be done by chaperones. So this chaperones are otherwise called as heat shock protein. It is otherwise called as heat shock protein. It is otherwise called as heat shock proteins. So you have two different heat shock protein, HSP60 and you have HSP70. You have two different heat shock proteins, HSP60 and HSP70. So what happens is you're going to have this protein folding. So this is going to go for protein folding. Once protein folding is done, there is going to be a protein targeting. That is going to be called, that is going to be protein targeting. What is protein targeting? It is going to be acting like your Amazon. So Amazon என்ன பண்ணுது? So உங்களுக்கு செல்லர் கிட்ட இருந்து ஒரு பொருளை வாங்குது, வாங்கி அமேசான் என்ன பண்றாங்க, அது அமேசான் வச்சு அதை வந்து உங்க கஸ்டமர் கிட்ட விற்கிறாங்க, கன்சியூமர் கிட்ட. So this is going to be your seller. From your seller, it has to travel to your consumer. So that is going to be done by your protein targeting. He is going to dispatch the consent target destination. This is what happens in the post-translation. What happens is, unfortunately, this process can be, what happens is, can be inhibited at certain points. So these factors can be inhibited at certain point. They have given you some examples. Proteolysis. So what is going to be example? Pro-insulin to insulin. Hydroxylation, proline and lysine in your collagen. Glycoxylation, carbohydrate addition to form glycoprotein. Gamma carboxylation is going to be done by glutamine, vitamin K, phosphorylation, serine, threonine, and formation of disulfide bond, 2 cysteine residues, methylation, lysine, arginine, histidine. It's the example histones. So the histones, you know, right? So the one which is going to bind your DNA is that one. And acylation is going to be myelin proteolipid or transferrin receptor. Now you have certain, there are certain inhibitors. Number one, which you need to understand is, so you have certain things that are going to be present in both eukaryotes and your prokaryotes. There are certain things that are present in the eukaryotes. There are certain things that are present in the prokaryotes. I'll just put a table. I'll just write it down. You just copy it down. So what happens? Now we are going to talk about inhibitions of protein synthesis. Okay, inhibitions of protein synthesis, guys, remember. So first let's talk about the prokaryotes and then we will talk about the eukaryotes. First put the heading as prokaryotes. Okay, so in prokaryotes, you're going to have number one, you're going to have puromycin. Then you're going to have tetracycline. Then you're going to have your chloramphenicol. So this puromycin, then you have this tetracycline, then you have chloramphenicol. Even they are used as some drugs. So on the drugs and the antibiotic use or antimicrobial. What it does is, it is going to go and inhibit your proteins in the protein synthesis of your bacteria. Thus it is going to die. So how it does? So the puromycin, they are going to form peptidyl puromycin. So a peptidyl transferase prevent panadu, and they are going to inhibit your translocation inhibit panna elongation liye cut aidum. Aduthathu, what your tetracycline does is, your tetracycline is going to block your A site. Therefore, the binding of amino acid tRNA is going to be blocked. Chloramphenicol enna panudana, they block your peptidyl transferase. They inhibit peptidyl transferase. Okay, and next one, what we are going to discuss is streptomycin. What is your streptomycin? Here is okay. What it's going to do? It is going to inhibit the initiation at high concentration. It's going to inhibit the initiation inhibit panadu. And what does your erythromycin does? So erythromycin. So what is the role of this erythromycin? Okay, what is the role of this erythromycin? The role of erythromycin is they are going to be binding to 50s ribosomal subunit, 50s subunit, and they are going to inhibit your translocation. They're going to inhibit your translocation. Okay. So this all are going to be the one that done in the prokaryotes. Prokaryotes in the inhibition aachna adu namakku nalladhu. Okay, prokaryotes inhibition aachna that is going to be good for us. There is no problem. But if the same inhibition occurs in the eukaryotes, if the same inhibition occurs in the eukaryotes, that is going to be danger. Now, eukaryotes எப்ப நீங்க drugs கொடுக்கலாம்னா when you have this two things. One, or bacteria is going to do something to us, or the second thing what can occur is you have this so-called anti protein synthesis drugs இருக்கு. அந்த drugs இருக்கலாம். But நம்ம மெயினா பார்க்க போறது வந்து bacteriaஸோட வேலை தான். Number 1 is going to be your diphtheria toxin. Diphtheria toxin. The diphtheria toxin என்ன பண்ணுனா they catalyzes ADP ribosylation of residue. So ADP ribosylation of residue in your EF2. And what does your ricin do? Ricin is going to inhibit or inactivate 60S subunit. It is going to inhibit 60S subunit. Similarly, you have the cyclohexamide. Cyclohexamide. What is the cyclohexamide going to do? It is going to again block your peptidyl transferase. It is going to block your peptidyl transferase. At the end of the day, what you need to understand is transferase. At the end of the day, what you need to understand is, so prokaryotes, this generally includes protein synthesis in the mitochondria and chloroplast. Okay, at the end of the day, what you have to understand is very simple, that prokaryotes நம்ம கொடுக்கிறோம், eukaryotes அது நமக்கு கொடுக்குது. So ரெண்டுமே டாக்ஸின் தான், ரெண்டு பேருமே ஒருத்தர ஃபைட் பண்றோம். So ஓகேவா, at the end of the day, that is what you need to understand. Now there is one more thing what you need to understand. I told you something called as protein folding. Now what happens is, this protein folding at this particular point, this protein folding can be having some disorders. So these disorders are called as protein misfolding disorder. You call it as protein misfolding disorders. So what are this protein misfolding disorders? I'll just write it down. Okay, what is protein misfolding disorder? Let me tell you guys, very easy, very simple. So number 1, what you have to understand is you have the so-called Huntington's disease. Huntington's disease. This Huntington disease is due to the abnormal folding. So what is that abnormal protein called as? Abnormal protein is called as Huntington. Okay, second one is going to be your cystic fibrosis. Cystic fibrosis, you all know what is the abnormal protein synthesized there, CFTR. So we call it a CFTR gene. Third one, remember Parkinson. Third one you're going to remember is your Parkinson. Parkinson, you're going to have alpha-synuclein. And number 4 is going to be your Creutzfeldt-Jacobs disease. Do you remember this Creutzfeldt-Jacob disease? You would have read in your somewhere in your biochemistry protein synthesis. You would have read in the protein. But anyway, if you haven't read your protein, once again, we are going to deal it in this biochemistry journey. First, we will complete the molecular biology. Then we are going to anyway move forward and talk about all the biochemical chemistry topics like your metabolism of your protein, metabolism of lipids and carbohydrate. In this order, we will be going. Okay, there we will be reading Creutzfeldt-Jacob is nothing but prion disease. We call it as prion disease. What is this prion? Remember, prions are proteinaceous infectious material. It's an infectious material. And it is no DNA or RNA. So there is no DNA or no RNA in this. And presol. So this can lead to certain disease that is called as your prion disease. Okay, number 5, you are going to talk about this Alzheimer's. You all know Alzheimer's, isn't it? Alzheimer's is going to be due to beta amyloid. It is going to be due to beta amyloid. Last and final topic which we are going to deal with is the targeting. So I told you one thing, right? Targeting. So what is this targeting? So it is Amazon, like I told you already, it is like Amazon. So once you receive a material, it has to send to some person who is required. So that you divide this into post-translational modification, and your co-translational pathway. I told you this also, translational pathway. Post-translation is going to occur in the nucleus. It is going to be occurring in the mitochondria. it is going to be occurring in the peroxisome. It is going to be occurring in the peroxisome. Whereas co-translation is going to occur in the Golgi apparatus. It is going to occur in the endoplasmic reticulum. See, in Golgi apparatus, what happens is, it is going to do this sorting and packaging work. It is going to do the sorting and packaging work. What it does is, if it adds, okay, there are two things. If there is a tag, okay, so if there is, if it is, what it is going to do is, it is going to add a tag called as mannose. Just this is an example. It add a tag called as mannose 6-phosphate. So the one who adds this tag, mannose 6-phosphate, the one who adds this tag is nothing but glucnac phosphotransferase. Glucnac phosphotransferase. Okay, he is going to add the mannose 6-phosphate. He is going to add the mannose 6-phosphate to the protein. If at all this mannose 6-phosphate addition to the protein is not occurring, if this is not occurring, then what happens is, this is sent to the, okay, this is sent to the ribosome. So what happens, it is then sent to the ribosome, and the ribosome is going to have this disease, and this is called as eye cell disease. So this is going to be present in the endoplasmic reticulum. What you have is this eye cell disease. From the Golgi, it is sent to the endoplasmic reticulum, and what you have is so-called eye cell disease. Just remember this. Then you have this protein targeting. What is targeting? The synthesis of protein on your endoplasmic reticulum, rough endoplasmic reticulum. A signal sequence of protein interacts with a single signal recognition particle, and SRP is going to be going on the RER, that is rough endoplasmic reticulum. The growing polypeptide passes through your translocation in the rough endoplasmic reticulum lumen, where it is going to get glycoxylated. So in the rough endoplasmic reticulum lumen, what happens? It is going to get glycoxylated, and what it does, it is going to get out. Okay, so this is how your targeting of your protein occurs. Now, can anyone answer me? Triplex DNA is formed because of? Triplex DNA is formed because of, remember, triplex DNA is formed because of Hook-Steen pairing. So this I didn't tell you, just remember this, Hook-Steen pairing. So for one peptide bond formation, how many high energy phosphate is needed? 4 high energy phosphate is needed. Which of the following is not true regarding eukaryotic gene? Non-coding intron contain nuclear gene and pseudogene, correct. Modification of mRNA occurs before transportation, correct. Non-coding intron is there, correct. But you have a monocentric mRNA. Polycistronic mRNA is not present in the eukaryotic gene, it is present in the prokaryotic gene. Once you understand all these things, next topic is the molecular biology technique, which we will be dealing it tomorrow. For today, you are going to understand this transcription very well. Okay, thank you.